This photograph establishes a terrifying baseline from the height of the epidemic. A diagnosis used to mean a rapid, inevitable, and fatal decline. Today, medical science has built a highly effective defense. Antiretroviral therapy, or ART, stops the virus from replicating, driving it down to undetectable and untransmittable levels. But maintaining those undetectable levels requires absolute adherence. Patients must take a strict regimen of medication every single day for the rest of their lives. Miss a few doses, and the virus bounces back. It's a permanent standoff where the medication holds the virus hostage, but never actually eliminates it. Which brings up an obvious question. After 40 years of intense study and billions in funding, why can't we just flush the remaining virus out of the body? The answer lies in a biological sanctuary known as the latent reservoir. HIV targets the immune system's CD4 cells. The virus injects its genetic code directly into the host DNA helix and goes completely dormant. Because this dormant DNA isn't producing anything, current drugs wash right past it, blind to the threat hiding in plain sight. As long as those dormant strands remain intact inside the DNA sequence, the infection survives. Reaching them is the only way to cure the disease. To reach those hidden strands, researchers are turning to a specific engineering tool, CRISPR-Cas9. While traditional drugs try to poison the virus from the outside, CRISPR operates on the inside. It acts as a set of molecular scissors that scans the DNA, locates that dormant viral code, and physically deletes it from the host cell. This mechanism is already moving out of the lab. Researchers from Amsterdam University and Excision Biotherapeutics are actively testing this approach in human clinical trials, known as EBT-101. The Phase 1 results prove the treatment is safe. In one specific case, a trial participant was able to stop their daily medication entirely and remained virus-free for 16 weeks before it returned. That 16-week window demonstrates what researchers call a functional cure, a state where the body's modified system keeps the virus controlled without any ongoing medication. The EBT-101 trials confirm a critical proof of concept. Locating and deleting latent HIV from a living human cell is a demonstrated mechanical reality. The immediate logistical obstacle is scale. The human body contains millions of infected cells scattered across different tissues, and the CRISPR scissors have to be delivered to every single one of those hiding places. To solve this, scientists are packaging the CRISPR instructions inside lipid nanoparticles and sending them through the bloodstream. That specific mRNA delivery system is exactly what was optimized and deployed globally during the COVID-19 vaccine rollout. But even with efficient delivery, it might be mathematically impossible to snip out every single piece of viral DNA. Scientists need a contingency plan for the cells they miss. Researchers at Johns Hopkins University are developing a parallel strategy called block and lock. Instead of extracting the virus, this method modifies the cell to permanently seal the viral DNA in its dormant state. The genetic code stays inside the host, but it is locked down so tightly it can never wake up, even without drugs. Whether the approach is to cut the virus out or cement it in place, these techniques share a common target. Biotech is taking the fight directly into the reservoir itself. Medicine is navigating the boundary between managing a lifelong disease and establishing a permanent functional cure. Crucial clinical data from these parallel approaches is accumulating right now, with major trial milestones scheduled for 2026. Over the next 5 to 10 years, researchers will focus on extending that 16-week window of remission, using these gene editing tools to iterate on the successes of the first human trials. For the millions of people currently living with the virus, this work represents the first viable path toward a life that doesn't revolve around a daily pill bottle. HIV's greatest strength was its ability to stay hidden. We are finally entering an era where we have the tools to illuminate the dark and target the virus at its source.